So hello everybody and welcome back to the eSchool of the European Society for Cell and Gene Therapy. And after you have heard in the last two lectures about lentiviral vectors, we are now moving on to the adenoviral vectors. And we really have here an expert on adenoviral vectors and oncolytic adenoviral vectors. It's Len Seymour, who is giving the talk today. He is a professor of gene therapy at the University of Oxford. And his science is dedicated on targeted cancer therapies. And there also, that's very important to work with adenovirus. Today, you have to be aware that we really have to stop a 1:45 because, as you may have noticed already, we will start today also with our e-seminar on SARS-CoV. Len, the floor is yours.
Thank you, Hildegard. Thank you, everybody, for joining today. I appreciate the opportunity to tell you what I know and what I don't know about adenoviruses. It's an interesting opportunity to talk about the breadth of adenoviral vectors, and I'll try to do it in as informative a way as I can. I'm conscious that there's far too much to talk about in 45 minutes. So I'm going to try and finish in 35 minutes. That means I will go quickly over things I don't understand and over things that maybe are so well known that they don't need to be explained. And I hope I can leave some time for questions at the end. So the title I was given was Adenoviral Vectors, Gene Delivery, Powerful Weapon in Cancer and Beyond. And I should disclose at the outset that I do own and I was a founding scientist in Cyoxis. And that's relevant because the very final slide is a Cyoxis slide. Okay, so the things I'm going to talk about today are, first of all, a little bit about adenovirus structure and biology, just the background, where I learned most in putting this together. I'll talk briefly about adenovirus for gene supplementation therapy. Then I'll talk about adenovirus for vaccination, particularly virally vectored vaccination. And then I'll wind up by talking about oncolytic adenoviruses. This is the overall structure of the virus. It's a double-stranded DNA virus. It is a human pathogen. Causes respiratory infections, conjunctivitis, urinary tract infections. Crucially, it's non-enveloped. And I really prefer to work with non-enveloped viruses because I think that they are easy to manufacture or relatively easy to manufacture because they're not limited in their manufacture by the amount of envelope available on the packaging cells. Now, I can't prove that, but I think it's probably true. And one of the major strengths of adenovirus is that you can make enormous batches of it. You can get 10 to the 13 virus particles per liter relatively easily. So it's easy to manufacture. And if you're looking for something that's going to be a drug, you might want to bear that in mind, that the cost of goods ultimately could be important. It's also non-integrating, which is a big strength because it's not known to cause cancer. The biology is fairly well understood. I wouldn't say fully understood, but the coronavirus epidemic is telling us that virus biology is never really understood. So adenovirus is relatively understood. And as you all know, there are packaging cells which contain key adenovirus genes, E1, which can be used to transcomplement replication deficient vectors to produce gene therapy vectors. So that's the structure of the virus. It's about 100, 105 nanometers diameter. It's got sticky outy bits called fiber proteins on the outside, fiber knobs, which are used to bind to high affinity CAR receptors. And then the peripentanal hexons bind also to RGD. It also binds heparin sulfate proteoglycans. It binds factor 10. It infects cells via a variety of different routes. These are some of the phylogenetic concepts of the virus. There are 57 accepted human adenovirus serotypes, and they're divided into 7 different species. Now, if you want two adenovirologists to fight, ask them how to divide the 57 human adenovirus serotypes into species, because some people think you should do it by cross-reactivity of antibodies, and that's predominantly what's done. Other people think you should take a more modern view and look at the genetic structure and categorize them that way. So you will see that on the right, the breakdown of the different structures is shown. The ones at the top, now it should say that this phylogenetic tree includes both human and chimpanzee adenoviruses, which are all part of the same family. The ones at the top of the group B adenoviruses, I'll just point out to you AD4, which is what, 97%, 99% identical with AD16, but is categorized in a different group. So AD4 is group E, whereas AD16 is group B. And you'll see that the categorization of these things is still controversial. Adenovirus is trying to sort them out, but it's still a matter of some discussion. At the bottom on the left, you can see the different respiratory and other types of diseases that the virus cause. And then it's worth emphasizing when we talk about some adenoviruses that there are different genotypes within the same serotype. So, for example, my favorite adenovirus is Ad11P, which means the prototype Ad11, whereas the other genotypes that have been discovered with the same serotype are given different suffixes. So here's something that I hope you all know, which is that if you look at the composition of, or you look at all the different adenovirus serotypes, and you look at the presence of neutralizing antibodies in the sera of patients, sera of healthy individuals, you find that some adenoviruses are very heavily recognized and others hardly at all. And Ad5, which is the one that most people have historically worked on, is probably the most common virus in our environment, and nearly everybody has got high levels of antibodies against it. Whereas other serotypes, and my favorite, as I said, is Ad11, but Ad34, Ad35, Ad43, have very low levels of antibodies in the circulation. Just something to bear in mind for later. Now, adenovirus is regarded as a relatively innocuous, relatively friendly virus. It's thought to cause respiratory infections, heavy colds, as I said, other types of infections. But given half a chance, it'll kill you. And this is something to bear in mind when we're using it in high amounts in people who are undefended against it. And you can see that this is true because in immune suppressed individuals like transplant recipients, they very often show a reawakening of adenovirus, either from their own tissues or from the graft. Nobody's quite sure if it's from the host or from the graft. But when they're fully immune suppressed, adenoviruses can reawaken. They can give rise to the regular infections that we see. And then in about 10, maybe 20% of those patients who are getting infections, it can go viremic. And that means it spreads around the body and you get circulating virus in the bloodstream. And that's a very bad sign because it can lead to severe pathology. And as we all know nowadays, there aren't very good antiviral agents. So cidofovir is the agent of choice as an anti-adenovirus agent. And about 2 out of 3 patients respond to it, but 1 in 3 don't. And there is an appreciable rate of mortality from adenovirus reawakening in transplant patients. Something to bear in mind, it's a friendly, well understood, innocuous virus, but it's not completely harmless. What I like about it is that I think it exemplifies molecular nanotechnology at the supreme height. It's a fantastic nanomachine. So here is a very simple diagram of the virus structure, attaching to high affinity and then low affinity receptors, which can vary depending on the cell type, pulls itself into cells, we think by a process of endocytosis, enters the early endosome, then group C adenovirus leaves the endosome relatively quickly, group B leaves it later. But in both cases, that process seems to involve a shedding of the fibers from the surface of the virus so that you end up with exposed hydrophobic domains on the vertices, which we think are the protein 6 being exposed. That will then melt through the wall of the endosome into the cytoplasm of the cell, and then it shuffles along microtubules to dock itself, so the capsid docks itself at the nuclear pore. And then a miracle occurs. This is something that very little work is done on. The genome of the virus appears to enter into the nucleus, and I suspect that it relatively efficiently traffics its way to transcription factories, or maybe it sets up its own transcription factories. It certainly docks to the chromatin, and it probably docks to the chromatin via these terminal proteins at the end of the linear genome. When it gets into the nucleus, it undergoes an immediate step of transcription, and it produces what we call the early proteins, or the early RNAs and the early proteins, which are involved in overtaking the cell's biology to allow the virus to control the cell and to amplify the DNA genome of the virus. So once the DNA genome is amplified, then it goes into its late phase and starts to express all the structural proteins from the virus, which are then made into capsids, which can package the virus for assembly and shedding from the cell. Now, the whole process from here, from attachment to nuclear activity, is thought to be about 40% efficient. It's the only estimate I've ever seen, but it's a really efficient agent. 40% efficiency for something that complex is impressive. And then for each virus that gets into the cell, you can get something like 10,000, maybe 50,000 daughter copies of progeny virions being released from the cell. So it's a fantastic agent. I won't dwell too much on the transcription side of it, just to say that the early genes, I mean, the virus is beautiful. It It carries its genes in both strands of its DNA. Some read from left to right, some from right to left. The early genes, the E1 genes, are clustered here at the left-hand end. The E4 genes are clustered on the right-hand end in the reverse orientation. E2 is down here and E3 is up here. And E means early. So these are the 4 early clusters of genes, E1, E2, E3, and E4. So those are produced early on in the virus life cycle, as I've said. There's then a magical switch that occurs, which is the activation of a thing called the MLP, the major late promoter. Something leads to triggering of the major late promoter, which happens after the DNA is manufactured, and that then triggers expression of all the late proteins. So transcription then occurs from the major late promoter to make the RNA, which is then spliced into different RNA sequences that encode all the different late proteins of the virus. So it's a simple virus once you understand it, but it provides us tremendous opportunities for engineering. And I wish I was talking about engineering today, because this is a virus you can make jump through hoops. It will do everything for you if you handle it well. In terms of vectorology, there are several different approaches you can use. You can think about using wild-type viruses, and I've thought about that many times as an anti-cancer agent. You can use conditionally replicating viruses. So this, for example, is CR2 deletion mutant of E1A, which means there are 24 base pairs taken out of the protein E1A. And that means it's no longer able to bind to retinoblastoma protein in the cell, and it doesn't put the cell into cycle. So that virus, which we call the Delta 24 virus, is dependent on pre-existing cell cycle. But you can also put promoters into the E1A gene, into the E1 region, or into the E4 region, and make it dependent on specific transcription factors, so you get cell type specific activity. So there's a variety of things there which are used in the context of oncolytic viruses, and I'll talk about that later. You can make replicating non-infectious viruses, and this is just one example with the deletion of the capsid protein P3A, which means that when this virus gets into cells, it produces loads of DNA, produces loads of proteins, but it doesn't produce the P3A capsid protein, so it can't assemble full viruses. And this is of interest as a vector for vaccination, because it's causing a pro-inflammatory effect from the virus replication, but it doesn't produce live viruses. So if you have a transgene in there, you may get a good transgene response. And then there are replication incompetent viruses, first, second, and now third generation. The first generation is deleted in E1 generally, normally E1A and E1B. There's also deleted in E3. Now, virologists took out E3 because it didn't really matter very much, but E3 is the part which regulates the viral defenses against the adaptive immune system, and to some extent the innate immune system. So viruses that miss this are not able to defend themselves against the immune system, and when they go in vivo, there may be an issue with that. Second generation is missing E1A, E1. It's also sometimes missing E2A and E2B. These are all agents that can be provided in trans during packaging, and they make more space to put in transgenes into the virus. And then nowadays, people are working with gutless or gutted viruses, which are missing the whole genome, and which can carry up to 35 kilobases of transgene in them. Problem with those is that they're relatively hard to prepare, and that's an area where they really ought to be more focused, because they've got tremendous potential in gene therapy. These things can be used in gene therapy, gene supplementation, or killing. In cancer, we use it as a strategy for killing cells. It can be used in vaccination. I'll talk about those later, and we can talk about virotherapy, so where the viruses kill the cancers. I can't really talk about adenovirus without mentioning this event, which was a tragic event which led to the death of a volunteer, Jesse Gelsinger, on a gene supplementation trial that was performed at the University of Pennsylvania. This was probably the single most high-profile event in gene therapy and caused enormous problems for the field. Jesse Gelsinger, I think, did more for gene therapy than anybody else, in the sense that it was this event which stimulated the formation of the British Society for Gene Therapy and did a great deal to galvanize scientists to realize we had to control our science better and we had to represent it better. So he died as a result of an enormous dose of Ad5 that was given into his hepatic artery to try and treat his ornithine transcarbamylase deficiency, which is a deficiency of the liver enzyme. Sadly, he was admitted to the trial, even though his liver function didn't appear to be quite up to it, and he suffered a massive inflammatory response, and he died. Now, with the benefit of hindsight, it's very easy to to criticize, but I would say that at the time, the field felt that this was a good trial to be doing. And now it's very easy for me to say the strapline at the bottom and say, well, this is a lifelong disease, and adenovirus is a non-integrating and therefore short-term expressing virus. So why would you use it for treatment of lifelong diseases? I think we've learned a huge amount from this, and we've now moved away from genetic diseases using adenovirus. We move more towards situations where we can express it for a short term for cancer treatment or for vaccination. This is an example of gene supplementation for cancer, where you deliver the wild type P53 gene. Now, P53 is the most, well, I think it's the most mutated tumor suppressor gene in any type of cancer. It's very heavily mutated in lung cancer, but in many others. I'm sure you all know it. It's what we call the guardian of the genome. So when P53 is mutated, and both copies have to be mutated, then cell is not able to go into cell cycle arrest and is not able to apoptose as a result of damage. And the concept was, if you restore functional P53, then you may restore the ability of the virus of the cell rather to undergo cell cycle arrest, and also to undergo apoptosis. This was a trial that was taken forward, an idea that was taken forward by Shirin Plow in the US. And the results of their phase 1 clinical trial were very impressive. This was in people with advanced ovarian cancer, intraperitoneal administration of the virus, and you'll see that the patients who were treated did far better than the patients who were not treated. And as a consequence of this, it's a long story, but a consequence of this led to the first Chinese acquisition of a drug that was immediately licensed in China. And this was, at the time, I think, the first gene therapy that was licensed around the world. It was only licensed in China, but worldwide, it was the first one to be licensed, to my knowledge. And people like me were saying, well, it's a mad idea because you can't replace P53 in every single cancer cell. And yet the efficacy is better than you would expect. And there must be an immune system kicking in here somewhere. But I think even at this stage, we don't fully understand it. TNF is another example of an agent that can be delivered using a virus to mediate an anti-cancer effect. Now, there isn't time to talk about this, but TNF is a very powerful anti-cancer agent. As a single agent, it's curative in mice, but given to humans, it was causing lethal toxicities. So Genvec thought, well, I wonder if we can deliver this in a virus. And sorry, I should say also, it's nowadays is used for isolated lymphofusion, given into the femoral artery and to treat advanced melanoma, sometimes sarcoma. And the results are quite stunning, but you can't let it go systemic because it's too toxic. So the idea was, can you express it from a virus and have it mediated local anti-cancer effect in the virus? And this was done. TNF was put into AD5. It was injected intratumorally into patients with head and neck cancer. And it was actually controlled with a radiation-inducible promoter to make it even safer. And the results were stunning. These are patients in the phase 1 trial treated IT with the drug called TNF-arade, given with fluorouracil and hydroxyurea. And of 12 patients, 5 underwent a complete response, 5 had a partial response, 2 had stable disease, and only 3 had progressive disease. So an 83% response rate. So fantastic results. The company then got excited, I think, and took it forward to phase 3 trials and went into pancreatic cancer, and it completely failed in pancreatic cancer. And I think there are many lessons in there, but one is that the right application, the right setting, adenovirus has got a lot to offer. But if you expose it to the wrong type of cancer and the wrong therapeutic strategy, it may be struggles. Then I want to talk briefly about adenovirus in vaccination. There are many different adenoviruses strategies that can be used in vaccination. And the one that I think we all think about is the one shown at the top here, where we take a replication incompetent adenovirus and put a transgene, this is an antigen transgene, into normally the E1 region of the virus. That virus can then be used to infect cells and express high levels of the antigenic transgene. So that's the background to what we might call virally vectored vaccines. But there are also antibody display approach. There are also viral display approaches. So let me just say that this approach, the virally vectored vaccines, is supposed to be very good for T cells and to some extent B cells. But B cell vaccinations normally respond better to particles which have got antigen displayed on their surface so that B cells can be triggered by them. And there are so many ways a virus and an adenovirus can be used to do that. You can use hexon display by putting antigens into the hexon. You can use protein 9 display. You can use fiber display. You can use penton display. All of these will display your chosen epitopes on the surface of the virus particles and potentially make very good B-cell vaccines. And of course, you can combine that approach with the genetic modification to make arguably a very good T-cell vaccine. And you may in that way make a superb vaccine. Now, this is just a little bit of information about the virally vectored vaccines, which are very much in the news these days. So vaccination with in this case, the adenovirus vector, will induce an immune response against the pathogen antigen, but also against the vector. And for that reason, people who work with intractable diseases often think about using what we call a prime boost approach, which is to use the same antigen delivered in 2 different viral vectors. The mechanism of these things is not completely clear. Is it necessary for the adenovirus to infect antigen-presenting cells in order to get its transgene antigens expressed on the MHC1 of the antigen-presenting cell, or not? Can it work by cross-presentation? That still isn't very clear. Again, it's something that we can argue about at length. It's good that it avoids risks associated with the virulent pathogen, but it's limited by an inability to express huge numbers of pathogen antigens. Now, in the Jenner Institute here in Oxford, which is in the same building that we are, they make use of chimpanzee adenoviruses. And the reason for that is that basically chimpanzee adenoviruses are almost identical with human adenoviruses, but because they seem to be only prevalent in chimpanzees, humans have relatively low levels of antibodies against them. And the viruses that you hear about in the press nowadays are the Oxford viruses, a chimpanzee adenovirus called ChAdOx1, which is being taken forward for treatment of SARS-CoV-2. These are some data where they were treating people with an Ebola vaccine and found good antibody responses, which was encouraging. And nowadays, you all know that these are the trials which have been completed and trials which are going on with chimpanzee adenoviral vectors worldwide. So there's ChAdOx1 down at the bottom here, there's ChAd3, and there's ChAd63, being trialed against diseases like malaria, HIV, Ebola, influenza, and TB. And then here we are with this virus, which is still out for testing. The first results in macaques were published and showed very good protection in the throat, some protection in the lung, and no protection at all in the nose. And it'll be interesting, and it caused decreased disease pathology. And we're very hopeful it will have a good effect in humans, and it's now entering testing in humans. And we will see how it goes on there. But you can see the background to this means that there are a lot of different ways that this agent could be potentially strengthened and improved. And finally, to wind up in the last, actually I'm going a bit too fast, and maybe in the next 10 minutes, I'll talk about oncolytic viruses. So this is a situation where oncolytic viruses are designed to infect and then replicate selectively in cancer cells. As I said earlier, you can consider using a wild-type virus, but most people, for safety reasons, use attenuated viruses that are deleted in one way or another to make them relatively safe. I may as well say here that I am not aware of any, well, I seem to remember in the back of my mind there was one situation, I can't pin it down now. I'm not able to tell you of any situation where we've had a pathology from an oncolytic virus replicating in humans. What we have seen a lot is toxicity due to particles being put into people, viral particles that just cause an inflammatory response. But viral replication in non-target tissues, I'm not sure I can give you an example of that. So you might consider wild type, but more often than not, people consider using attenuated viruses. Tumor cells can be then lysed as the virus replicates and the virus spreads on through to infect adjacent cells. It's designed that it will enter normal cells, but not infect them. And some of the strengths of this approach are that you can have an amplification in situ of the therapeutic agent, because you're putting in, in principle, one virus and you end up with 10,000. It can be highly selective, and I'll show you how in a moment. If you choose the right virus, it can cause a lytic cell death, which is a big bonus in cancer therapy, because lytic cell death is pro-inflammatory, releases inflammatory mediators like damage-associated molecular patterns, calreticulin, ATP. And you can also use it to express therapeutics. So you can combine oncolytic virotherapy and gene therapy. And that, I think, is a very powerful field. These are some of the viruses that are being developed in the clinics for oncolysis. So adenovirus was probably the original, but nowadays all of these different agents are in the clinics one way or another. And as you all know, the very first agent, the herpes simplex Defic, has been licensed in the US and also in Europe for treatment of advanced melanoma. I want to talk briefly about the archetypal virus in this space, which is Onyx 015. Excuse me. So Onyx 015 was designed to kill p53 mutant cells. And it was designed so that it couldn't defend itself against wild-type p53, but if p53 was present in a cell, then the virus was inactivated. And so the virus was attenuated by taking out E1b55K, which is normally the protein which binds p53 and targets it for degradation. But in the absence of E1b55K, then the virus is able to proceed fine in the absence of p53 in cells, but in the presence of p53, the p53 would either cause a cell cycle to inactivate or to cause apoptosis. So on the left here, you can see a wild-type cell. Virus gets in, causes activities that lead to an upregulation of p53, which then inhibits the cell activity and inhibits the virus activity. When that p53 is not present, however, you cannot get the cell defending itself, and the virus will then go on to achieve replication and lysis and spread to adjacent cells. Now, I'll say at the outset that this was not actually the mechanism of action, although it worked to some extent in the clinic. The mechanism of action turned out to be a transcomplementation of late RNA export and not directly what we thought it was, the p53 mutation status. So it was a complicated story, even more complicated than these graphs suggest. And these are some of the impressive data that were gained on that very first clinical trial 20 years ago now. In this case, the virus was given by direct intratumoral injection together with cisplatin and 5-FU into head and neck tumor nodules, and there were very good complete responses in injected nodules and not in non-injected nodules. And this is, for me, the most dramatic one. Just in passing, it's not very often I get to talk to so many people, but if you're making intratumoral injections, you need to be careful, because as soon as you take the needle out, everything drips back out the hole here. And you can see that even happens in the clinic. So you need to leave your needle in for a long time if you're making intratumoral injections. This agent did well. Onyx did well in its clinical settings. They then made a strategic decision that they were not going to take it forward any further. And the Chinese again bought the virus, and Shanghai Sunway Biotech developed it, or a very closely related version, as H101. Which I understand is still in use in China. Okay, so the basis for cancer selectivity. This is something that I've been wondering about for a long time. So these are the hallmarks of cancer, defined by Hanahan and Weinberg. And I thought it's useful to think about the hallmarks of adenovirus infection. Now, apart from angiogenesis and invasion and metastasis, all of these things have kind of equivalents in adenovirus infection. So, for example, activation of the cell cycle is something that the virus does as soon as it gets in. It needs the cell cycle machinery activated to allow the virus to go through its full life cycle. Inhibition of apoptosis is crucial, and this is also something that happens in cancer. So the phenotype of cancer can be complementing to a mutated virus, to an attenuated virus. I hope that point is obvious. I didn't say it very clearly, but what it means is that the phenotype of cancer can often provide what's needed for a virus to undergo its life cycle, even if the virus is attenuated in some genes. And we now know which parts of adenovirus are involved in which functions here. And so it's possible to make viruses that exploit different hallmarks of cancer. Now, we were thinking about this, but decided that we were too stupid to do it effectively. So what we did, and this is where the Sarxis work kicks in, this was working with Shearing Plough to work with the process of bioselection, where we made, with Terry Hermiston, a very large library of recombinant adenoviruses. So they were chimeric viruses, huge library of these things that we then selected for the viruses with the best ability to kill cancer cells. So this was done on the basis of most rapid lysis of cancer cells, and we ended up with a handful of viruses, which were then screened for the ability to kill normal cells. And we found two viruses which were outstanding in their selectivity, one of which has gone forward and is now known as N-adenotussirev. And this is the virus. We still don't understand its biology. We could never have designed this, to be honest. Missing big chunks of the E3 region. It's missing a component of the E4 region, which we think is involved in deregulating cancer cell metabolism. And it may be that that's a transcomplementing feature that happens in cancer cells to give it the selectivity. And there is also a chimeric region in E2b, which gives a chimeric polymerase between AD11 and AD3. Whatever happens there means this virus replicates really quickly. That's a very good polymerase that's been introduced there. Now, the reason we went forward with the one virus I mentioned and not the other was that it was on an AD11 capsid background, whereas the other one was an AD3 capsid background. And we know from the very first slide that AD11, not many people have got antibodies against it. So this virus, Adenovirus, has always been developed for intravenous use. And we now reach the stage where we think we can give the virus by intravenous injection and achieve infection of disseminated disease. And there the virus can mediate both viral lysis and gene therapy. So on infecting cancer cells will cause lysis and replication, but can also be used to produce therapeutic agents in situ. And the one thing I'm going to talk about for the last two slides is where we use the virus to produce bispecific T-cell engagers. This is for targeted immunotherapy. So bispecific T-cell engagers are these small molecules which cross-link a target antigen on a cancer cell with CD3 on a T-cell. And that means the T-cell is activated to kill the cancer antigen. Now here on the right, I don't know if I can run it back, but let me try. Here are blue T-cells killing cancer cells in the presence of an anti-EpCAM bite. You'll see that just a small number of T-cells is able to kill a very large number of cancer cells. They do this by recognizing via the bite the EpCAM molecule, then becoming cross-linked and then degranulating and killing the cancer cells. And the cancer cells, when they go green, they're apoptosing. So you can see that nearly all of the cancer cells are killed by that very small number of T-cells. Now, without dwelling too long on this, just want to say that I think T-cells are an incredibly powerful approach for exploitation by oncolytic viruses. And if you can use oncolytic viruses for specific activation of T-cells, and this is immunotherapy, so we're telling the T-cells exactly what to do, you can retarget pretty much any T-cell to kill any antigen-positive cell. And doing that within the tumor microenvironment is a tremendously powerful way to go. One thing I would mention here, because I seem to have got another 90 seconds, is that this allows killing of cancer cells independent of their HLA-1 expression. Now that's crucial because although we don't talk about it much, HLA-1 is lost in many cancers. Something like 50% of breast cancers have no HLA function. And it means that immunotherapy using regular T-cells against them is going to be a real challenge. The only type of immune therapy you could use is a natural killer cell therapy or a macrophage therapy or a T-cell therapy that kills via an HLA-independent route. And this, of course, is HLA-independent because it's just a bite binding it to an antigen on the surface. There's no involvement of antigen expression and presentation by HLA here. So I think this is a very potentially useful way forward. And this is a trial which SARCS is running, which is currently open in the US, and which was developed in our lab. So I can claim academic credit for this. The idea came from Kerry Fisher, and most of the work was done by Josh Friedman. The idea here is the virus enters cancer cell and expresses three things. It expresses a bite that targets CD3 to recognize FAP, fibroblast activation protein, which is present on tumor fibroblasts. So that means you're going to activate local T cells to kill tumor fibroblasts. And the idea here was, well, the virus should kill the cancer cells, but if you can get the T cells to attack fibroblasts, you may be able to do something about reversing immune suppression inside the tumor and repolarizing the immune suppressive microenvironment to make a more pro-inflammatory environment. And to help that, the virus also encodes alpha interferon, which activates dendritic cells, and also encodes CXCL9 and CXCL10, which are there to recruit T cells into the microenvironment. So putting all these three things together, we think it should be a very powerful way of creating a pro-inflammatory environment where the adaptive immune system is capable of killing fibroblasts independent of any HLA present. And the T cells should then be expanded. And of course, if they have endogenous selectivity for cancer antigens, which many of them do, but they're subject to immune suppression in the tumor microenvironment, then they may go on to mediate direct anti-cancer effects as well as anti-fibroblast effects. So very hopeful for this agent and looking forward to the first clinical trials results. So where does the virus sit in our overall armamentarium? Well, I'd say it's a relatively well understood virus family, and it's a very broad family. There are loads of different viruses you could choose from, each with slightly different properties. Most people have got one or two favorite ones, but there are many out there that are just not being explored. It's a double-stranded DNA virus with a proofreading polymerase, which means it's genetically stable. I do worry when you see people developing RNA viruses for oncolytic activity, putting very large doses. NDV is a reasonable example. Very large doses of viruses into humans, which we know are safe in humans because they're not human pathogens. But we were thinking about coronavirus as an oncolytic virus only a few years ago, and working with animal pathogens is potentially something it's very much something we should be considering the safety of. I'm not suggesting they'll jump species directly in humans, but they are known to do that elsewhere, so we need to be careful. As a human pathogen, adenovirus is unlikely to cause a pandemic. Versatile molecular biology, this for me is the crucial thing, because you can manipulate this virus. You really can make it do fancy things. We know such a lot about it, and the best part of my career is spent wondering how to make adenovirus perform useful therapeutic activities. Then it has limitations on its long-term applications because it doesn't integrate, but that means it's relatively safe. So it's useful for cancer gene therapy, virotherapy, and vaccination. And then the most crucial point of all, when we get something that's going to be useful, it's going to be easy to manufacture. So the Jenner talk about making vaccines at $1 a dose. I don't think we'll reach $1 a dose for anti-cancer agents, but it's an interesting concept that could be achieved with this vector if we really put our minds to it. Thank you very much for your attention. I'm very happy to take questions, and I'm hoping to hand back to Hildegard now to chair. Thanks, Hildegard.
Thank you very much, Len, for this wonderful talk, the deep insight you gave on adenovirus and oncolytic adenovirus. And we indeed have a lot of questions already waiting for you. To those of you who were not there at the beginning, we have to stop at 1:45, but we will answer your question when you send this also to ESGCT office. So we start our question with Arjun. He is asking, what is the rate limiting step for adenoviral trafficking to the nucleus?
I have no idea. It binds to dynein and dynactin and pulls itself to the nucleus. I mean, the rate limiting step could be, there are so many points that could be. It could be escape from the endosome. It could be binding to the microtubules. It could be entry of the DNA through the nuclear pore. I mean, frankly, I don't know, but I would, if I had to bet on one, I would think it would be entry into the nuclear pore, because that is something that we understand very little about, and I would imagine it's very complicated.
So Margarita is asking, what is the modern understanding of the cell death type caused by AD, especially by species C AD?
That's a wonderful question. Something that we think about a lot. So type 5 adenovirus contains a thing called the adenovirus death protein, which is expressed late and appears to induce an accelerated death over and above what would be caused just by lysis. Other viruses, like group B viruses, don't have a death protein, and so they may die in a slightly different way. You can argue that activating apoptosis makes no sense for an adenovirus because cell death by apoptosis could well activate DNases that could be damaging to virus genomes. And I've seen no real evidence that adenovirus activates apoptosis. Apoptosis tends to be something that smaller, less sophisticated viruses use. So the evidence that I've seen suggests that adenovirus is killing cells by a process either of lysis or of there's a process called oncosis, which is energy deficient cell death, very similar to lysis, but It's not a coordinated death pathway. It's not necroptosis, not apoptosis. It's like lysis, but it's a little bit more regulated, and it causes production of very large single membrane blisters from the surface of the cell. Now, the videos we've got, but they've been with group B adenovirus, tend to suggest that cell death is mediated in that process, which is, as I said, a process called oncosis. With type C adenovirus, I think it's probably more like necrosis, but it's not something I can give a definite answer to.
Thanks. So Arjun is asking, is there a tissue-specific tropism for ad vectors known?
Well, in the wild type, largely it's defined by their ability to find cells. So adenovirus is pretty good at infecting most cells that you feed to it in the test tube. Obviously, when it's exposed to humans, it tends to be a respiratory infection because we breathe it in. Is there a tropism for infection? Well, some of the species give more urinary tract infection. So group B adenovirus is infected by CD46 and desmoglein 2, as opposed to group C adenovirus is infected by Coxsackie adenovirus receptor. I've always thought that one of the more important things is going to be the accessibility of the different receptors on the surface of cells when you're using a virus to treat them. Because what you do in a test tube is you tend to work with cells that are either not very polarized, or if they are polarized, then they tend to be, at least my understanding is, they're polarized upwards, so the apical face is towards you and the basolateral surface is away from you. And we see pretty good infection with most viruses in those settings. But actually, if you work with semi-permeable membranes and polarize the cells on that surface, you can find that there is potentially differential ability of some adenoviruses to infect from different surfaces. Now, this is one of the reasons I'm interested in group B viruses, because they infect very well from underneath, at least in our hands. And of course, if you're going to use a virus by intravenous injection, then it goes into the bloodstream, and when it extravasates, it's coming to the epithelial cells. If they're polarized in the cancer, it's coming to them from the basolateral surface, not from the apical surface. So you need a virus which is going to be able to infect from that surface. Now, I'm not saying that this is hard and fast science, but I am saying that it's something that we really should consider, because just using cells in vitro probably simplifies the system too much to make it translatable.
The next question have been asked by two different persons, and this is, are there any bystander effects after adviral therapy to surrounding non-infected cells?
I'm not aware of any. The best data we have is mixed cell populations in vitro using, for example, endothelial cells and tumor cells. There we can see that treatment with an oncolytic virus will completely destroy the cancer cells, and it seems to leave the endothelial cells unscathed. Now, we've never checked to see that all of the endothelial cells are still there, but you certainly see a lot of them, and they don't look particularly unhappy. So I'm not aware of any bystander effects in that setting. Of course, if you had other epithelial cells, particularly cycling cells, you might expect, depending on the structure of the adenovirus that you're using, you might expect to see some pathology of the virus in those cells. But it's not something that we've seen in our own experiments, no.
So I have two questions from Margarita for you. One is, what is, in your opinion, the best place to insert transgenes in the E3 region for producing effective oncolytic vectors upstream or downstream of ADP? This is the first one. You may already start answering.
I have no idea, Margarita. I know people that would have a view on it, and if you want to contact me, I can put you in touch with them. But no, I have no idea. Sorry, too complicated.
The next one would be, do you think the in vivo selection of ad library of different serotypes is a good approach?
Absolutely, yes. Yes. So in vivo selection using relevant model systems has got to be the way forward. And libraries, I'm quite convinced that libraries will allow us to find much more sophisticated approaches to treat cancer than we could design from our own brains. I mean, we still don't understand the selectivity of N-adenotussirev, and we would never have designed it but large recombinant libraries and relevant model systems has got to be the way to go forward.
And there's a further question of her, which says, how effective are advectors expressing anti-CTLA4?
Good question. There has been data on that in the literature now for about 15 years. CTLA4 is the most sensible antibody to express, or anti-CTLA4 is the most sensible one because it's so toxic when you give it intravenously. The idea of producing it locally within tumors is a great idea. All I can say is it's a great idea, and there are data out there in the literature that suggests that it can be successful, but I haven't seen any test in clinical trials.
The last question I have for you is from Ivan. He says or asks, what are the best ways to purify a viral drug for clinical application?
Well, for clinical application, it normally has to be done depending on the jurisdiction that you're in, but most jurisdictions like to keep away from cesium chloride gradients. So in the old days and in the laboratory, even now we use cesium chloride gradients, but it's generally done by affinity chromatography or by aqueous two-phase chromatography or some other physical technique that allows selection without contamination. I think affinity chromatography is probably the most reasonable. But this is a very big field, and many different production centers around the world are trying to develop new strategies for production. And most of them are protected and patented in secret. So my answer is a very primitive one. Chromatography is probably the best, but I appreciate that you probably have a much greater insight into it than I do.
So that was my last question for you, Len. Thanks again. It was a beautiful and wonderful seminar. And for all those who have now further questions, please shoot them, send them to the ESGCT office. We will send them to Len for answering. Thanks again.
Thank you, everybody.
And see you next week. Bye-bye.
Bye-bye.